This experiment demonstrates how to utilize mass spectrometry to monitor hydrogen-deuterium exchange associated with protein structural changes that lead to activation states. First, cleave protein kinase PAC2 with caspase 3, and then activate PAC2 by autophosphorylation using ATP as a substrate. As a second step, digest PAC2 with pepsin and analyze the proteolyzed PAC2 fragments by tandem mass spectrometry. Proceed to analyze hydrogen-deuterium exchange patterns of PAC2 by MALDI-TOF monitoring of the deuteration level of each PAC2 fragment. The precise level of hydrogen-deuterium exchange using MALDI-TOF analysis provide important insights into caspase 3 cleavage and autophosphorylation of PAC2.
This method can help answer questions of protein dynamics, such as protein conformational changes, protein-ligand interactions, and protein-protein interactions. The main advantage of this technique over existing methods, like HD exchange LC-Q mass spectrometry, is instrumental. This procedure does not require an HPLC-linked mass spectrometry to analyze HD exchange results, and the procedure is easy to operate.
To cleave 0.6 micrograms of PAC2, add caspase 3 and incubate the reaction at 34 degrees Celsius for 30 minutes. Verify PAC2 and complete cleavage of PAC2 by analysis of products by SDS page. For the activation reaction, add the cleaved PAC2 to 20 microliters of activation buffer.